Let's get the video. Let's see who's here. Right? So for you viewers at home, there's going to be a row of cells here that's not got either one of these in it. Now, so that's shown here. In that particular row of cells, for these two, particularly these two, Eve and Futz, those two, this is Even is the name of the full name, and this is Bushichiratsu. Yes, it means something in Japanese like every other or something like that. But there's going to be one row. That particular row, that particular cell that's in that stripe, that would be located right along about here, is going to express wingless, the gene wingless. And the cell immediately to its posterior is going to express the gene engrailed. Okay, so wingless we've heard of before when we talked about signaling pathways. Wingless is the fruit fly version of the gene we call Wnt. And the gene Wnt is called Wnt because it's a combination of a vertebrate gene and the fruit fly gene that are the same gene. So wingless in fruit flies is the same as a gene that was identified in mouse called INT1, Roman numeral INT1. So this, the obvious phenotype is a wing defect. The wings don't form properly. But wingless is not just expressed in the wings. So the wingless mutation that has improper wings is not a complete loss of function of wingless. It's actually a loss of regulation of wingless. It's a mutation not in the protein, but in the promoter or the enhancer that keeps it from being expressed properly when it's supposed to be making wings. But it still makes all these stripes. If you didn't do this, you'd never have a fly, right? So it's expressed early during embryogenesis, but it's misexpressed later, and you get no wings. And that's how it got its name, wingless, right? This one is a gene that was discovered because the insertion of a virus in mice always happens, or almost always happens, or very frequently happens in this gene's, in the region of this gene, and screws it up. And as a result, that can often cause tumors to form in the mouse. So this was what was thought of as an oncogenic virus. It was actually murine, mammary, and leukemia virus. So it would cause basically the mouse version of breast cancers and the mouse version of leukemias. And it was, it's a retrovirus type virus, and it gets itself integrated into the genome. And one of the places where it often gets integrated, they called site of integration 1, and they just named that region INT1. Well, it turns out that when they identified this protein, the gene, it encodes a protein that is basically identical to wingless. It's a secreted, it's a cell signaling molecule. So they said, all right, let's make one name, wingless and Wnt. We can kind of pronounce that. So the Wnt signaling molecules are a combination of the wingless protein in fruit flies. So wingless is Wnt, and this protein in mice. And there's wingless in fruit flies, but there are numerous Wnt proteins in vertebrates. Okay, I don't know, we're up to like 11 or something. There's a bunch of them. So wingless is going to be expressed here. What about engrailed? Engrailed is a transcription factor. It's usually a repressor. So it binds to DNA and it represses transcription. But it also can activate its own genes. All right, so this is going to cause a border to form between these groups. And this border represents the cell right at the posterior part of the parasegment, but is pretty much right smack dab in the middle of the segment. And so wingless is going to be expressed and secreted. It doesn't go too far. It only goes about 3 or 4 cells in each direction. Okay? So in order for it to function, it's going to need to bind to its receptor. And its receptor in fruit flies is the gene frizzled. And frizzled activates the protein disheveled or somehow modulates that protein. And disheveled, when it is so modulated, will interact with the fruit fly protein called ZW3 or ZEST-white3. So you never actually say ZW, you say ZEST-white3. But ZEST is a weird consortium of mutations in fruit flies. And then it's got all these other things tagged onto it. And we'll see another ZEST in a minute. But ZEST-white3 is a kinase, and its job is to phosphorylate a protein called armadillo. Armadillo, when it is normally being phosphorylated, armadillo will be destroyed. Okay, so when this is being signaled, disheveled is blocking this, and this arrow means armadillo will accumulate. Okay, and when it accumulates, it goes into the nucleus as a transcription factor. So this is the Wnt pathway. We have looked at it earlier. We didn't use all these names. We used the names that everybody else uses now, which is Wnt will bind to frizzled. Frizzled is the name for everything still. Frizzled will activate disheveled, which is the name for everything, but it's based off the fruit fly. Disheveled, in turn, will inactivate what most people will call GSK3. Okay? Glycogen synthase kinase, which is a gene that was originally just thought to just do stuff in metabolism to get glycogen, sugar, metabolized or released and let go from the liver or muscle cells or wherever you're storing the glycogen, right? Well, it turns out to have kind of another role, and it's a pretty important role, but GSK3 equals ZEST-white3. And this ordinarily would repress the protein we would call beta-catenin, which in fruit flies is named armadillo. Okay? Beta-catenin would ordinarily be involved in cell adhesion. These are cadherins, and you have on the inside, you have beta-catenin, then directing interaction to the cytoskeleton, right, in both sides. And that lets the cells anchor together at an adherens junction. Great. So beta-catenin is really involved in cell adhesion. But beta-catenin can also, if it's just hanging out in the cell, can go on in to the nucleus and regulate some gene expression, okay, and cause some gene expression. Well, under normal circumstances, so if Went's not there, Frizzled's not doing this, Disheveled is not doing this, GSK3 is phosphorylating beta-catenin and destroying it, okay? So when beta-catenin is phosphorylated, it's destroyed, okay? But if you inhibit GSK3, then GSK3 can't inhibit beta-catenin, right? So how do you inhibit GSK3? Well, you activate Disheveled. How do you get that done? Well, you activate Frizzled. How do you get that done? Well, you bind it to a receptor. So Frizzled is there on the cell membrane, activates Disheveled. Disheveled inhibits GSK3, and the way it inhibits it is it just complexes it into a bunch of other proteins, so it can't interact with beta-catenin. So now you don't phosphorylate the beta-catenin, so now it doesn't get destroyed. And sure, there's some over there doing the cell stuff, but it was there anyway. But all the extra beta-catenin goes into the nucleus, turns on a bunch of genes. It's going to turn out that that's pretty important because this is how you got your backbone. Okay? This is why you have a back and a head. Well, in fruit flies, it's how you get a stripe. Okay, so wingless protein activates this pathway. So it activates Disheveled, which then inhibits SWITE3, which then allows for armadillo accumulation in the nucleus. And armadillo activates a bunch of genes like engrailed and hedgehog. Hedgehog is another secreted signaling molecule. Hedgehog is even less soluble than wingless, and so hedgehog only signals to a few cells in its nearby vicinity. And its receptor is called patched, and there is no patched on the cell that expresses hedgehog because, remember, engrailed is a repressor. Engrailed actually represses hedgehog. So over here in this cell, you have engrailed, and it's repressing hedgehog receptor, which is patched, okay? So it's repressing the receptor, which means hedgehog is made here, but hedgehog can't signal here, nor here, nor here, because everywhere where wingless goes and hedgehog doesn't, engrailed gets turned on, and engrailed turns off the receptor for hedgehog, but everywhere that was already expressing patched and wingless gets to respond to hedgehog and gets to make a bristle, okay? Gets to make a bristle, or gets to regulate how its bristles are made. All right. So you're going to get a gradient of wingless that's expressed from the posterior border of the parasegment to high to low towards the anterior border of the parasegment. And you're going to get a gradient of hedgehog that's high at the anterior border of the parasegment to low towards the middle of the parasegment. Okay? So in the middle, you kind of don't have either thing signaling much. At the anterior parasegment, you get hedgehog. At the posterior, you get wingless. These are secreted proteins. Right? This is cell signaling. What was the molecular function of the maternal effect? This is sort of like, this would be a great exam question. What is the molecular function of the maternal effect gene? How do they work? That's what they do, but how do they do it? What's their molecular function? How do they work biochemically? Okay, so they're transcription factors. They can repress or activate. A couple of them are translation repressors too. Don't forget that. Then you have gap genes. What is their molecular function? Or activate transcription factors, right? So they're repressing or activating transcription. So transcription factor is a molecular function, right? Like the types of the actual biochemistry that they do. That's what we mean when we say their molecular function. Okay, what's their developmental function? That's the answer you gave about specifying polarity. Okay, so molecular function is how do they actually act? What do they bind to, and how do they do it? So transcription factor. What about the pair rule genes? But, Eve, what?
Well, it says it regulates segment polarity genes.
That's what they do developmentally. How do they do it? They're transcription factors, right? So if you regulate a gene, you're a transcription factor. All right. Here is the first point we get to where we have something that isn't a transcription factor. So what's the molecular function for hedgehogs and wingless? How do they carry out their job? Signaling factor, right? So they're a secreted ligand. They're activating signaling cascades. So the direct thing that they do is bind to a receptor and set in action a signal cascade that goes through, right, transducing proteins like disheveled and GSK and beta-catenin to eventually affect transcription, yes, but they don't directly affect transcription, right? They're not binding to DNA, so they're a signaling molecule, right? What's the molecular function of frizzled? Receptor. What's the molecular function of disheveled and GSK3, right? Transducers, right? So what's beta-catenin's molecular function? Well, it is an odd protein because it has two molecular functions. One, it can serve as a transcription. It's actually a co-activator. It doesn't bind directly to DNA. It binds to a protein that is, but it's a co-activator of transcription. And it's a cell adhesion controlling protein, right? A long time ago, there was this Saturday Night Live skit. You may have Googled it at some point, come across it. They used to have really good commercial skits on Saturday Night Live. And so this husband and this wife are having an argument, right? And they've got this can, it looks like a whipped cream can. And he's like putting it on his ice cream. And she's like, what are you doing? He goes, I'll put it on my ice cream. It's a dessert topping. She goes, no, that's a floor wax. And she sprays it on the floor and she's mopping with it. And Dan Aykroyd comes in, it's both. It's a dessert topping and a floor wax, right? How moronic, right? So that's what I always like to say about this, which no one gets, but now you will. Beta-catenin, it's a dessert topping and a floor wax. It's a transcription factor and a cell adhesion molecule, which are very different classes of functionality. Just they shouldn't be in the same ballpark with each other, but they are. All right. So that interaction of wingless is going to set off through beta-catenin a whole bunch of gene expression in the posterior parasegment, and hedgehog signaling is going to set off a bunch of gene expression in the anterior part of the parasegment. And those two gradients are going to create patterning, and we can see that in the larval structure of denticle bands. So there are four major sort of clusters of these denticles or regions that either have them. So the primary denticles are these pretty large ones that are right at the highest hedgehog expressing area. And then you have a zone that's actually barren of denticles. And then you have a tertiary zone that has smaller, slightly differently shaped denticles. And then you have a broad zone with very fine, thin denticles. And then again, right at the end here, where the highest wingless is, you'll have a semi-clear zone of denticles, followed by a single row of cells expressing denticles immediately at that hedgehog boundary. So that patterning is very reproducible, and the width and the size and the extent of those denticles in each segment is very reproducible. So this is a blow up of the third segment there, third abdominal segment, to show you those denticles, and they form in the cuticle of the fly. And this particular patterning is the top part of the dorsal side, and it's different on the ventral side. On the ventral side, you have these denticles being mostly over here, where you have high wingless, and not so much over here where you have hedgehog. Okay, so if you look at the bottom part of this, it's kind of the opposite, right? So you have lots of denticles up here, not so many down here. In the mutation hedgehog, the reason it's called that mutation is this signaling isn't happening, and instead you have the wingless signal throughout. And what does the wingless cells have on them? Lots of denticles. So you have denticles everywhere. The thing is really bristly looking, like a little hedgehog has all of its little bristly hairs, right? That's how it got its name. So hedgehog and wingless, wingless we just saw is length. Hedgehog is hedgehog, okay? So in mammalian systems, we have hedgehog. We call it sonic hedgehog, or Indian hedgehog, or desert hedgehog. But we have still hedgehog. Most people don't even try to distinguish between sonic and desert and Indian. They just call it hedgehog, just like in fruit flies. And we have the equivalent of patched, and the other gene, which is called smoothed, which is a co-receptor, and so on and so forth. So the hedgehog signaling pathway is conserved. The Wnt signaling pathway is conserved from fruit flies to you. All right. So this brings us then to our final group of genes. We have the homeotic genes. So the gap genes and the parallel genes initiate stripes and define borders. Ultimately, the segment polarity genes establish the anterior posterior of each individual segment boundary. But how does the boundary between T1 and T2 and T3 and A1 and A2, how do those segments actually know, hey, I'm supposed to make a leg here, or a wing and a leg, or a haltere, or an antenna, or an eye, or whatever is going on in the abdomen? Well, that comes through the homeotic genes, right? So the gap genes and the parallel genes initiate the expression of the homeotic genes. They're some of the primary drivers. And then there's another group of genes that I'll introduce in a minute called polycomb and trithorax genes that will modulate them as well. Okay. Once you have the homeotic genes begin to be expressed, the segment polarity genes sort of dictate what they do within that segment, help to dictate the more refined expression pattern within that segment. A lot of times the images are drawn and it's like, okay, the entire thorax segment 1 has sex cones reduced in it. But actually, it's not. It's restricted within that segment, and it's restricted by segment polarity genes to a degree. Okay? So all of these classes of genes that we've talked about so far, the gap, maternal effect, pair rule, and then ultimately the segment polarity, all interact with the Hox genes to activate them and create a boundary of their expression. Once the Hox genes begin to be activated, they self-regulate. So many of them will activate themselves and repress their neighbors that would be expressed on either side to create very defined boundaries of expression in each region of the embryo. So that's where we would say that the homeotic genes are somewhat auto-regulatory. They auto-activate, and so there's a feedback activation, and then there is inhibition by their neighbors, from their neighbors in adjacent segments, or adjacent regions, which define those segments then. Okay, does that make sense? So it's a compilation. That's why we started this whole thing off with a hierarchy of gene expression. Okay, a hierarchy means it's like you've got, you know, president and this and this and this on down, right? President of the club, vice president of the club, and then you got the, you know, the flunkies that are doing the heads of different committees, right? So the party committee, the cleanup committee, the flyer distribution committee, potluck committee, so on and so forth. And there's a chairman for that. And then there's the committee members. And then within it, basically, you get to the lowly little people that are just doing the basic tasks, right? Or think about it like in the Army. You got a general and then whoever comes after that. And then finally on down to the basic grunt that's doing the actual fighting. So you have that hierarchy of this gene expression as well, where each level sort of communicates with the next level and directs the next level and on down until ultimately you have the instructions that say, you're going to make a leg right here. You're going to make a wing right here, okay? There are 8 homeotic genes that you need to know the names of and the order that they're in, okay? And the order is important because the order that they are in is the order in which they are expressed through the embryo and adult. So their names are labial. Just the abbreviation is here, but labial means lip, right? This gene is expressed in the mouth part segments. Proboscipedia, that's a little tougher. Proboscis, that's like means nose, right? It's also expressed in parts of the mouth part segments, right? Insects have like 5 segments that make up their mouth: maxillae, mandibula. What's that?
Pharyngeal?
No, there's the 2 parts that are on the side, and there's 2 parts that are on the top. But anyway, proboscipedia. So pedia means leg. So this mutation is named for the effect, which is when this gene is mutated, the mouth part segments, some of them, turn into what look like legs. So the mouth segment turns into a leg instead of a mouth. That's a homeotic transformation. Homeotic means a identity or common origin, right? Homologous and so on. Homeotic means you have transformed one part into another part. So the mouth is transformed into a leg, okay? The next gene is deformed, like you would think, spelled, deformed, and it affects this region in the head. And the third gene is called sex combs reduced. Sex combs, like you comb your hair, reduced. On the front leg of fruit flies is a structure known as the sex comb. It's a row of about 8 or 9 bristles that they have right here, only on males. And they use that to rub the bristles on the female's wings as the mating ritual. That's how the little guys kind of, go up to the little girl flies and they start rubbing their wings. And if the female is receptive to mate, then she smacks him with her leg, and then he turns around and mates. Okay, so females don't have them, but males do. And when this gene is defective, the sex comb doesn't form on males. Okay, But in reality, the reason the sex cone isn't forming is because you don't have a front leg. You have two middle legs. So the front leg turns into a middle leg, which wouldn't have a sex cone on it. But the middle leg and the front leg have a whole different set of bristles. So if you look closely at the leg, you go, oh, that's not even a front leg. That's a middle leg. They've got 4 middle legs instead of 2 fronts and 2 middles. Okay, the next gene is called Antennapedia, which is the name for this entire cluster of 5 genes, Antennapedia complex. Antennapedia. Oh, antenna, which is normally controlled by labial, as well as some of the mouth parts. Antenna is a leg? Yes, that's exactly what it means. The antenna turns into a leg. Then there's these are all on chromosome 3. Then there's actually a very long stretch of the chromosome, a big window where there's a whole bunch of other genes. And many of those other genes are actually members of this family called homeotic genes, or Hox genes, but they aren't involved in homeotic transformations. So you skip a ways and you get to 3 genes that are called the bithorax. It means 2 thoraxes, because when these genes are mutated, especially this one, ultra bithorax, put an ultra in front of that, ultra bithorax, you get 2 sets of wings. Rather than just 1 wing on the middle segment, you get a wing on the 3rd segment. You're not supposed to have a wing there if you're a fly. It's good if you're a dragonfly or a butterfly or a wasp or a bee or an ant. But if you're a fruit fly, you're not supposed to, or a mosquito or a housefly or a gnat. So that means you get an extra thorax. So that's ultra bithorax. And then the last 2 are abdominal A and abdominal B. They should all be capitalized. I'm not really sure why they didn't capitalize. So abdominal, right? That's just because it's in the abdominal segments. So here's a little schematic of the embryo at full germ band extension when the segments have been defined at this point, and there's 14 of them, and you can see the expression, except they left off Proboscipedia because Proboscipedia overlaps with both of these. That's fine. It's kind of one that's a little weird, but gives you these things. And then here's SCR, and then Antennapedia in a couple of segments, and then ultrabithorax, and then several of them, and abdominal A is in all these, and abdominal B is just in the last group. So they're expressed in more than one segment, but they're not expressed equally in those segments. Each gene in the posterior segment negatively regulates the genes to its immediate anterior. So for example, Antennapedia has a negative regulatory effect on sex cones reduced, restricting sex cones reduced to that segment only. Ultrabithorax restricts Antennapedia to that segment, or those 2 segments only. Abdominal A restricts ultrabithorax to primarily the 3rd thoracic segment, and abdominal B makes abdominal A be expressed most highly in the 1st abdominal segments. But it's not 100%, but it's a higher versus lower expression. All right, so what are the 8 homeotic genes? Labial, Proboscipedia, deformed, SCR, Antennapedia, ultrabithorax, abdominal A, abdominal B. Okay. So you do learn them in their order. So the way they were initially discovered is that people would look for mutations or found mutations where one structure was completely changed over, and that called that a homeotic transformation. So for example, Antennapedia, where the antenna become legs, that's pretty freaking noticeable, and it's pretty freaking awesome, which is this guy right here. Okay, so normal fly. Here's your mouth parts, right? There's 3 or 4 of them. Here's the antenna, and this is the arista, and here's the eye, and so here's antennapedia. Yeah, that's not an antenna, okay? It's not a really good leg either, and it doesn't work as a leg. They can't walk on their heads, but it is, yeah. So you'll also notice, what's that? That's not a normal mouth part either. That also looks like a little bit of a leg. So in Proboscipedia, only the leg forms here, but in Antennapedia, actually legs always actually form in both places. Okay, this is not a good thing. Okay, so this mutation is actually dominant, and actually in most forms it's lethal. Okay, there's only a few forms of it that are not lethal. And when you want to say, oh well, I want to look at a fly that's got Antennapedia, I've bought some before that have Antennapedia, and you look in the bottom and you'll get, well, none of these have legs on their heads. That's because the little boogers knock them off. They break off. Okay, they're not supposed to have this thing dangling off their head, and it gets broken off. And they're heterozygotes, because if they're homozygous, they die. So you have to breed them like every 2 weeks. You have to remake the whole group and sort out the Antennapedia virgins, and yeah, it's a pain in the butt. Somebody did it, but not this guy. He's not doing it. So it's cool to see them. You get, like, if you get a culture of them, there'll be like 2 of them in there that have legs on their head until they're about 1 day old, and then the leg gets knocked off. So you can tell they don't have antennas, but you can't tell that they have a leg. So it's cool, but it's not really as cool as it might sound. Oh, going the wrong way. So the other really noticeable mutation is ultrabithorax. So look at this guy, man. So you have an extra shield structure here, scutellum, that's like you would normally have on the second thorax, which kind of covers up the first thoracic segment. And you get another one, and you get wings. No, they really can't fly very well, okay, because they're not designed to fly with 4 wings. A dragonfly can't, right? So if you look at a dragonfly, its second and third segment each have a pair of wings. So do butterflies. So do wasps, bees. The wings are stitched together with little bristles, but it looks like 1 wing, but there's really 2 wings. Flies, what is their family name? Or Insecta? Yeah, family, right? Order. Diptera, 2 wings. So they're not supposed to have 4 wings, but ultrabithorax does. Well, the segment that normally makes the wing is the segment where Antennapedia is supposed to be. So Antennapedia is not supposed to be here, but it is, because ultrabithorax isn't working and keeping Antennapedia where it's supposed to be. So Antennapedia is actually getting to be expressed in the 3rd segment of the thorax, and it goes, I'm here, I'm going to make a wing. So you have a wing. I'm not supposed to. So those 2 mutations are actually 2 different categories. So Antennapedia, that causes the antenna to form, is really considered a gain of function, right? And that results in what we call a posterior transformation. A posterior transformation is a little tricky. A posterior transformation is when an anterior structure transforms to look like a posterior structure, right? So posterior getting moved and transforming the anterior one. That's a posterior transformation. It's a gain of function. So Antennapedia was expressed in the head, which is where it's not supposed to be, because it gained that function. Okay? So gain of functions make anterior things look more posterior. Loss of functions, just the opposite. they make posterior parts look more anterior. Okay? So if antennapedia is supposed to be restricted by ultrabithorax, and ultrabithorax is lost its function, what happens? The anterior function gets to come back. So you have an anterior transformation. This more posterior segment looks like its anterior neighbor. That's because UBX is lost its function. Okay? In this mutation, antennapedia is functioning fine as a protein. It's just functioning in the wrong place. In this mutation, antennapedia is also functioning fine. It's simply functioning in the wrong place. So this led developmental biologists to realize that most of the changes that are probably happening with organisms are not really due to mutations in the function of a protein, but due to mutations in the regulation of the expression of that protein. So you have antennapedia just being antennapedia, but it's being antennapedia in the wrong place, maybe, or at the wrong time, maybe. And that's going to cause a big difference in structures, right? That's a pretty big change in the structure of the fly, especially this one, and it has huge evolutionary consequences or sort of overtones, because you think, well, it's been long thought through insect evolution that the flies, the dipterans, are sort of like far down the evolutionary scale. They're the most recent developments, right? And organisms that had 2 pairs of wings would represent less or earlier evolutionary branches on the tree of insect development. So basically what you're doing is you're reverting a fly back to sort of a previous or earlier evolutionary form that insects had when they had 4 wings instead of 2 wings. And that's kind of cool. It has some cool implications. And in fact, if you look at organisms that are arthropods that have like a shrimp or something, right? They have little appendages on every abdominal segment. They have legs on their thorax, and then they have appendages on every abdomen, right? All the little swimmerets that you've ever eaten shrimp, you peel them off, right? Or a lobster, anything like that. Well, why do they have all those little things? And insects don't. Dragonflies don't. Wasps don't, right? They have an abdomen that doesn't have any appendages. That's an insect. Well, if you look at the antennapedia gene and you look at the ultrabithorax genes, those 2 genes have distinct differences in their sequences. And if you take, for example, the fruit fly type of antennapedia gene and mutate the shrimp version of that gene to look like the fly version of that gene, you lose those structures on the abdomen. So it's like antennapedia is being expressed in those organisms in a different way in the abdomen and making little legs, swimmerets, on their abdomen. And if you can block it, then you get a shrimp with no swimmerets, which would be awesome, right? Especially if it's a crawfish, because it'd be easier to peel and you could eat the crawfish easier, right? I'm saying. So anyway, these homeotic mutations are really fascinating and really cool. On the other hand, they're very deleterious to the organism, but they're really cool to us, for us to think about. All right, so we'll keep talking about that on Monday and then get into dorsal ventral, and then we'll probably be finished with all that on Wednesday. We'll give us Friday to kind of talk and sort. Remember, Monday we're gonna talk about your research plan for next week's lab. All right? Sound good?